I think you'll really like. So let's just look at the title: Long-term transmission of defective RNA viruses in humans and Aedes mosquitoes. So the virus we're talking about here is called dengue virus. And so, yeah, you know dengue virus. And dengue virus type 1 is the most common and I think the most dangerous version of dengue virus that's out there. And so this is transmitted by mosquitoes in the genus Aedes. So Aedes aegypti, I think, is one of the main ones that does, but there are other Aedes that do transmit it, I believe. And so so we got those parts. We know that Aedes mosquitoes normally transmit dengue. That's fine. And so we're transmitting it to humans, in humans. Great. Okay, fine. And so long-term transmission of defective RNA viruses. What does that mean? Well, this means viruses, so dengue is an RNA virus, but these are viruses that don't actually encode a working anything. They are defective. They have parts of the genome, but they don't have enough of the genome to actually work. Let me show you what that means. So if we were to go to viral zone over here, let's go to viral zone, there we go. Then we could look at, let's see, they're positive stranded, single stranded RNA virus. And let's see, we're looking for dengue, which is going to be a flavivirus. Here we go. And so it's going to be in the flavivirus genus. And there we go, dengue virus. Fantastic. So the virion looks like this. It's got a really neat fusion protein. Well, the fusion protein is going to start out laying down like this. And so these are actually two proteins that are laying down and forming a homodimer. Actually, each one is two different versions of the protein. But they are going to stand up when they get fusion activated. And instead of dimers, they're actually going to form trimers. So two from this one and one from that one will form up. And one from this one and two from that one will form up. And they'll just stand up like a little teepee, and they'll be dimers. Now, this is the bit that I wanted to look at. So this is what the genome looks like. So dengue virus is sort of like poliovirus. It only encodes one great big gene, which is another one of these polyproteins, which just means it's got all the proteins, but they come out joined together in one big piece. And so they would look something like this. And everywhere that there's a little arrow, this is some kind of protein cutting enzyme that will actually cut these proteins apart. And so you can see it's a mix of signal peptidase. This is something that's found in the cells that's doing all the dark blue cuts. And the Golgi protease, there's a protease in the Golgi that cuts right there. There's a protease that we still aren't sure exactly which one that cuts between NS1 and NS2A. And then down here, the polymerase is there, and most of the cuts are going to be made by NS3, which is this sort of aqua-colored thing, and that's going to cut everywhere that there's a little aqua-colored arrow. And so, yeah, that's what they look like. And so can you imagine what would happen if you took a protein and, let's see, the E is the yellow one, that's the one that's going to be on the outside, the E protein. If you stopped halfway through the E protein because there was a mutation that just ended the open reading frame, well then, you could make the core protein, the C, and you could make the pre-M and the M, which is those two, the orangey ones, and you could start to make the E protein. You could make maybe this much of it, and then you would stop, which would mean you wouldn't make the NS3 protease that cuts here and all these other places. You wouldn't make NS4, which is involved in sort of anchoring the complex, the replication complex, and you wouldn't make NS5, which is the polymerase. So you would basically not have a virus if you stopped there. But that is exactly what is going to happen in this study. All right. And so these are all from Myanmar, or a place that used to be called Burma. And this was done by looking inside of people who had dengue fever virus in them and reading out all the genomes to see exactly what there was. And what they found was this. They amplified out all the E genes, and remember E is something like the fourth gene along in the little chain. There we go, right there. And they noticed when they made a family tree that it grouped up like this. So this is inside of different people. You could have, 10, 20 different strains going on in there at once. But they found that people had a large number of strains in this gray box. And all these things had a stop codon that broke the open reading frame. And so these things could not make a full-length E protein and couldn't make any of the proteins downstream of that. And so the idea here is that these viruses are dead, but because of the way quasispecies works, the strong viruses, the regular full replicating viruses, and these guys can get into the same cell at the same time. And if they're in the same cell at the same time, the working virus will actually copy these guys, and so they get a free ride. They get to come along. But it's not like they're doing nothing, because these are going to build up mutations, because of course now they're no longer tied to fitness. They don't have to be able to copy themselves. They're parasites of another virus. And what this does for the whole virus quasispecies is that all these viruses are having to compete against the host and against each other. But these guys can build up mutations, and mutations are usually bad, but if you get enough of them, you can sometimes get to a place where those mutations would not be bad. And when that happens, then you can sample parts of this genome and transfer it back into the other ones. And so you can make giant evolutionary leaps going from fitness peak to fitness peak without ever having to go through the valley in between. So viruses cooperate, and even the dead can continue to evolve when you're part of a quasi-species. Now, how neat is that? I think all the way neat. Anyway, this will be one of our papers to please have a look at. And we'll read through and see if you get it.